Hi, Assalamualaikum. This is our biology case study. The title is Micropropagation Plant Cell Culture, BIO270. And for our members, the first one is NAWWARAH binti NIZAMMUDDIN. For the second one is SARA EMELIN FARISHA binti MUHAMAD FARID. And the third one is BALQIS AQILAH binti MOHMAD NOOR. For content, we have introduction, micropropagation's process step, strength of micropropagation, weakness of micropropagation, example of plants used in micropropagation, and conclusion, also reference. As for introduction, what is micropropagation? Micropropagation is the artificial process of producing plants vegetatively through tissue culture or cell culture technique. In this artificial process of propagation, plants are produced in vitro by asexual means of reproduction or by vegetative propagation. Plants can be produced both asexually via vegetative parts, multiplication, or asexual via seed production. One of the means of asexual reproduction is by multiplying genetic replicas of plants that are referred to as colonial propagation, wherein plants can be populated from a single individual through asexual means of reproduction. For the in vivo propagation of specific plants, asexual production via multiplication of vegetative part is the only resort, since they do not generate functional seed as seen in figs, grapes, bananas, etc. Successful application of clonal propagation to the following is observed is potato, apple, and many other ornament plants.
Micropropagation process step. The first step is initiation. Select a small piece of plant tissue, often an actively growing shoot tip or explant. Explant means tiny, sometimes microscopic, portion of plant material taken for culture, and sterilize it to remove any contaminants. Next, culture. Place the sterilized tissue in a nutrient-rich agar medium containing essential nutrient, plant hormone, and growth regulators. The tissue is cultured in vitro, allowing it to multiply. Usually, the medium will contain selected concentration of plant hormones, typically low or no auxin and higher cytokinin levels that induce good quality shoot. The next step is multiplication. Under controlled condition of temperature, light, and humidity, the plant tissue multiples, forming multiple plantlet. By altering plant hormones level, typically low or no auxin arrays and cytokinin level plus higher level of mineral supplements, shoot from the initiation stage are induced to proliferate into clumps of tiny shoot. These clumps may be subcultured or divided into separated shoot and placed in fresh media and separate containers. The process being continued until sufficient shoot have been obtained. Off type occur or the culture loses its ability to be subcultured. This process can be seen as taking cutting in miniature. The next step is rooting. When sufficient growth is achieved, the plantlets are transferred to a rooting medium with appropriate hormonal treatment to encourage root development. The shoot from the multiplication stage usually lack root until transferred to a medium that induces root formation. Rooting can also be achieved in more familiar horticultural soils cutting media, peat or coir mixture, for example, in humid conditions, as with conventional softwood cutting. The last step is acclimatization. The rooted plantlet are gradually acclimatized to the external environment to prepare them to transplantation to the field. Strength of micropropagation. The first one is rapid clonal propagation. Micropropagation produces genetically identical plant in a short time, enabling the rapid multiplication of valuable or high-yielding plant varieties. Next, conservation of rare species. It can be used for conserving and propagating endangered or rare plant species. Next, disease-free plant. The sterile condition reduces the risk of disease transmission, ensuring the production of healthy plants. The last one is year-round production. Micropropagation allows year-round cultivation and production, contributing to a consistent supply of crops.
For weakness of micropropagation, we have 4. Cost. Firstly is cost. The initial setup and maintenance of tissue culture facilities can be expensive. Second, labor intensive. Micropropagation requires skilled labor to maintain sterile condition and carry out the various steps. Third, genetic uniformity. While genetic uniformity can be an advantage, it can also be a weakness, making crops vulnerable to specific disease or environmental change. Fourth is stress-induced mutation. Some plants generated through micropropagation may exhibit genetic mutation due to the stress of the in vitro environment.
So next, this is the example of plants use micropropagation. Propagating plants that are hard to propagate, for example, orchid. The current low cost of this houseplant is due in part at least to the use of micropropagation of these slow-growing plants. Micropropagation of apple has played an important role in the production of healthy, disease-free plants and in the rapid multiplication of scions and rootstock with desirable tree. Tissue culture techniques are used worldwide to produce pre-basic, virus-free seed potatoes known as microtubers. For the conclusion, overall, micropropagation is a valuable tool for crop improvement and conservation, but it is essential to carefully manage its limitation and cost to maximize its benefit for food security and the economy in Malaysia.